You might only get a small amount of DNA from your DNA extraction. So how do you get more of that specific region of DNA you're interested in? You do this with the polymerase chain reaction, or PCR for short. The PCR can make millions and millions and millions of copies of the region of DNA you're interested in. This might be a DNA barcode, or it might be another region of the genome you're interested in, which is important for forensic analysis or for cloning. There are three main steps in a polymerase chain reaction. The first step is denaturation. We heat the sample up to 95 degrees Celsius, and this separates the complementary DNA strands. The second step in a PCR is called annealing. This is when we have a forward primer and a reverse primer. They're also called oligonucleotides, and they're between 18 and 22 base pairs in length. The primers stick to the opposite ends of your target sequence. This typically happens at a temperature between 50 and 60 degrees Celsius. The final step in your PCR reaction is called extension, and this happens at 72 degrees Celsius. This is when the Taq polymerase, a special enzyme, sticks to the end of your target sequence and extends along the DNA strand, making an exact copy of your region of interest. Denaturation, the DNA strands are separated. Annealing, the primers stick to the opposite ends of the target sequence. Extension, the Taq polymerase makes an exact copy of your DNA strand. We then repeat this cycle 30 to 40 times, and this is what makes those millions and millions of copies. So let's take a look at how we set up a PCR. Firstly, we have our PCR master mix. Inside the master mix, we have all the reagents we need to do our polymerase chain reaction. This includes a buffer, it includes our forward and our reverse primer, it includes our DNTPs, A, T, C and G, it includes magnesium chloride and Taq polymerase. Once we've got our master mix set up, all we have to do is add our DNA. And we mix that into the PCR master mix. And then we transfer it to our PCR machine. This is called a thermocycler. Thermocyclers come in different shapes and sizes. This one is really handy because you can use it out in the field or in the classroom. And it's really cool because you can actually control it using an iPad or a smartphone. We simply program in the different temperatures and the timing, we press go, and the PCR is off and running. PCRs normally last for about 2 hours.